Hello!
Hi there.
Welcome to Microbiology Journal Club, where we nerd out big about all things small.
My name is Danny, and in a previous life, I dropped out for my PhD in microbiology at the University of Chicago, where I was infecting skin I grew with MRSA. Nowadays, I'm a fact checker for pharmaceutical advertisers and the president of Biotech Without Borders, a nonprofit based in NYC dedicating to aiming the public with the tools of biotech.
My name is Faz, I have a PhD in microbiology, and I've mostly worked on bacteria, but I've also worked in publishing as a research integrity specialist, and currently I'm going to be working as an editor for a scientific journal.
Every week, we meet to talk about microbiology, and today it's our deep dive week where we look at, or sorry, it's our news week where we do an overview of papers that we've seen in the news or other sources to try to find an article that we want to cover next week in more detail.
Next week will be our deep dive week, where we take one of the articles we talk about today and we focus it figure by figure to learn what it can and cannot tell us. So make sure to subscribe to satisfy your microbiology curiosities.
You can follow along with any of the papers that we discuss in either week in our shared Zotero library linked in the doobly-doo below.
And we want to hear from you, so please use the comments or tweet us using the hashtag microtwjc hashtag. And boy, do we have a show today. We are looking at some really interesting stories. There's a new AstraZeneca vaccine targeted towards new variants. There's what's COVID doing in the brain, how is it triggering the immune system. More ways that SARS-CoV-2 can evade our immune system. But beyond that, is it possible to create a vaccine against all flaviviruses like Zika, dengue, and yellow fever? And also, have researchers found out a way to finally make phage therapy work? And also bees. I'll let
Yeah.
And finally,
Lots of different topics.
yeah, so we're going to be asking the real questions today, including how much does SARS-CoV-2 weigh? Stay tuned for all these stories and more.
And first up on the docket, we actually have something I guess that's close to our philosophy about things, this open science saves lives, lessons from the COVID-19 pandemic.
Yes, right, so for our first one, we've got, okay, yeah, so open science saves lives, lessons from the COVID-19 pandemic. So, all right, so this one caught my attention quite because, right, so, so we talk about a lot of things. So last week we did talk about, well, last news show we talked about how there's a lot of COVID research being published in preprint servers,
Yeah.
and that's so in terms of open science, that's kind of a good thing because we want to have every article that's about where researchers are working on COVID to be available, even if it's not very good. So we have this broad kind of scope of looking what people are doing. So I mean, open science is generally a good thing, but I think one of the things that have really helped us as a YouTube channel coming through is the fact that all these big publishers have made all their SARS-CoV-2 research available freely.
Yeah, because otherwise they'd be trapped behind paywalls, right? And I think we've experienced this a little bit. Like, there are papers that interest me that I'm like, oh, we should cover it on the show. But then when we follow the links, if they're not COVID-19 papers, then sometimes they're trapped, and that's hard to discuss them. And we say at the beginning and end of the show that we feel like what we're doing here is a type of peer review. I mean, it's not like the people working in the field, but we have some expertise. We're trying to continue a conversation to disseminate the ideas and also like think about them critically. And that's something that you can't do for some papers because they get trapped behind paywalls. And if you think about in different institutions, like not every institution has a subscription to every single journal out there. And so when publishers, I guess, made the decision to make COVID-19 papers mostly open access, I think that it was done because the thought was you could have more people looking at all these different papers, right, and then hopefully advancing that scientific conversation.
Yeah, and I think that releasing these preprints is quite a good thing for enhancing the scientific conversation. But open science isn't just about making sure everyone can read research. It's also making sure that everyone can, say, perform open source. So everybody has access to the same kind of tools and techniques to analyze data, and also open data. So rather than just looking at the graphs, actually being able to delve into other people's data to look at things, and also open peer review, where we can see the process by which something has gone from, say, a preprint to an actual published paper. something that I think we've touched on one or two times.
Yeah, I
Um,
mean, like we've seen some in the eLife, they have some of those reviews that are published. I see Nature Communications is starting to do it a little bit more. Yeah, little bits of it are peeking out here and there. And open data, we've seen examples of that, like just last week with the Cell Atlas. All of that is like an open
mm.
data set, right? And we even saw they made a tool to help people that don't have some of the experience in coding, like play around with those dots.
Yes, but the thing is, not this paper, this commentary delves into the kind of dark side of that. That it's not all rosy. There are, like,
Mm.
say, a lot of studies that have methodological weaknesses that limit the interpretability of their findings and can lead to false claims with a potentially dramatic impact on public health. So,
Hmm.
we generally don't like get in the business of debunking bad research because I think we try to promote our sciences. But last news show, I think I did make an example of like a paper that was shared as a preprint and then had hit a lot of news outlets. The one
Hmm.
way, which was actually had an interesting core to it on the kind of way that SARS-CoV-2 vaccines might be transcribed in the nucleus and what that might have, the effect that might have on splicing. And then
Mm.
the news stories the authors went with was, oh, this is the reason for the vaccine side effects, where they didn't actually have any evidence to link what they were working on that study to that outcome. And
Hmm.
so this preprint that arguably wasn't particularly great was suddenly being shared in news stories to say something that the data didn't seem to be actually saying. And
Hmm.
that's always been kind of the risk with preprints that because they're available for everyone, that means that everybody can read them, even people who don't necessarily understand them, and then that leaves something open to misinterpretation. Um,
Yeah, yeah. And so that's what this paper that we have in front of us is speaking about, right? Like specifically about preprints and how they're not peer reviewed.
mm,
And most preprint servers they do put the caveat on it, right? Like this isn't a peer reviewed source, but there's still that opportunity, I guess, for journalists and us even, right, just to pick up these papers. And if we're not careful to say like these aren't peer reviewed, and and we're not, I guess, technically savvy enough to look in those methods and identify the
mm.
potential methodological shortcomings, it could cause a situation where we're relaying messages about I mean, from the pharmaceutical advertising world, it's like about claims, right, that aren't
Mm.
really fully supported, or at least they're not being supported well by the literature.
Yeah, and this paper also delves into not only that question, but also the question of like the actual stability of the peer review, like because a lot of journals have implemented this thing called fast track peer review for COVID articles, where you people get deadlines of about 7 days to review articles, and
Mm.
there is a concern that reduces the quality of. I mean, yet another thing that comparable, like we give ourselves 7 days to review articles, so take that into account when listening to us sometimes. Um, but
Yeah,
yeah,
totally.
so I mean, they look into retracted papers particularly, and they find that like some of them were retract. Some of these ones that ended up being retracted had been peer reviewed in less than 3 days, and
Mm.
editorial conflicts of interest. And then they delve into this idea of editorial conflicts of interest. So the idea that people who are editors of a journal peer reviewing or like submitting their own articles to the journal that they edit for, and the finding
Mm.
that a lot of articles so whenever an article is submitted by someone who is on the editorial board, it seems to be reviewed much faster than articles that aren't submitted from the editorial review board. Um,
Hmm. Yeah, I think
so
that's figure two, right, in this paper. I
yes,
was
it is.
scrolling through a little bit.
Yeah, I'll scroll down to show people. Okay, figure two. Yeah, so like time to acceptance, like any editorial's
Hmm.
conflict of interest, they suddenly they get submitted in 1 day or less. So
Yeah,
they look at
accepted.
articles. Yeah. So basically, they look at two article types. There's reviews and editorials and viewpoints. So editorials, that's not so surprising because the whole point of an editorial is for an editor to say their views on a subject. So that doesn't
Hmm.
surprise me. Research articles does surprise me. But then there is another kind of factor that I don't think they account for is the fact the people from the editorial board are usually picked as high level people in their field. So
Mm.
when you recruit someone to an editorial board, you usually try and look at their kind of research record. You look at things
Yeah.
like H-index to check how successful they are. So the question is whether this measurement is picking out people who are, say, more successful, getting articles accepted faster, away
Mm.
from that association. So what I'd really like to have seen from a paper like this is a control for like something like H-index, and
Mm.
I think that even with that this is something that to keep an eye on, and something that, like, as someone who's worked in publishing, that we have to be. I never had a problem for my journals because my journals have had a criminally slow turnaround time. So
I mean, one
uh,
day or less, that's really quick, right? Like,
yeah, one day or less is
the
like
reviews would have to come. Is that what it's saying? Like the reviews and everything, the decisions just made in one day?
I assume it's submission to acceptance, which again
Yeah.
is incredible because I mean a lot of articles and journals I've worked with have sat in pre-screening for that time or longer.
Mm.
So where the basic things of have you written your article in English? Has it is there image duplications in there? Are you like uh, is this going to be a level suitable for peer review? I mean, getting something accepted in that time is incredible to me. Um,
Yeah.
but it's not unheard of. I mean, especially if say you're relaxing things in order to get COVID-19 research through. The sort of the publisher side steps would be the ones that would be taking the hit mostly because you'd recognize that people who will be able to review this are COVID-19 researchers who have probably got lots of other things they would like to be doing other than reviewing your articles.
Right.
So yeah, this it's so it's kind of interesting for that perspective. So it's interesting from uh, and there's also like another thing that has been a big part of the kind of research kind of discussions is the uses of press releases and
Mm.
uh, yeah and they also have a control. They look at the previous pandemic, so Ebola and Zika outbreaks, and they also notice that the actual research wastage, there's a problem of like lots of articles being published that aren't in preprints that never make it to actual
Mm.
publication. So
Hmm.
about like 48% to 60% of preprints don't make it to publication, and they
Hmm.
particularly highlight hydroxychloroquine because that story like that was
It
what made
blew
one
up,
it
right?
blew up. Yeah,
Yeah.
I mean
But
two.
then but then I guess after some of the more definite papers were published that shut down that theory, right, and the
Mm,
efficacy of it, then all those things that were sitting in the preprint world just waiting to have their chance, like they were no longer no one would want to publish them anymore, right? Because they had already sort of successfully been debunked by published papers.
yeah, I mean there were like 218 registered trials ongoing, and that doesn't count the many trials that were conducted without pre-registration, which again is registering a trial is a very important thing to do if you're doing something very important because it allows you to like be honest of what you're going to be doing in the study ahead of time, and so
Mm.
it's kind of essential for most clinical trials to do it. It's something so that the
But
research
it's not
community.
necessarily a bad thing. Like, do you think it's a bad thing that there's so much preprint research and then it doesn't get to publication?
I don't think that's a bad thing. I think that's
That's just part of the system, I would
yeah,
think.
I think that is part of
Yeah.
the system, and it kind of shows why journals exist. It's people don't necessarily can't necessarily like go through every single preprint, so journals are kind of important for that curation. I mean, but then again, I am kind of a very biased person as someone who's worked with journals. I
I
mean,
think to me, when I think about this, maybe what matters, I would think is like who's reading the preprints. If there's tons of these preprints and they're being read, then there's obvious utility there, right? Because it means that like other scientists are picking up these preprints and synthesizing all these little tidbits of information. But if they aren't being read, then you're kind of in this scenario where, well, people are showing what they've done, but no one's really paying attention to each other either, right? And so what is the value of just saying that we did this thing because later down the road, like that thing isn't important?
well, I mean, on the first hand, like studies like this actually show like or actually where preprints come in useful because then we wouldn't have know there's lots of people who've been working COVID-19 and we'd never know that they've been working on it except for the existence of these preprints. And so this
Yes, I
is
see.
actually so it comes in useful for studies like this which take a broader view of the literature. And also
Hmm.
in terms of sometimes preprints do get shared between communities without ever being published, but they're still useful because they're still part of that conversation. And
Yeah, yeah, that's what I was thinking. Like it's like I think that's a big part that I'm not sure everyone has absorbed yet in the world in terms of like what are preprints essentially, but they're this type of communication between scientists, right? Like there are these ideas in a not fully polished form, and they're intended for their colleagues, not necessarily intended for the public. And then, but that's where maybe some of these negative effects come into play, where they get amplified as if they were for the public.
I mean, I guess that brings us to figure 3, which shows like where the preprints are being shared, and the majority of them are being shared over Twitter. So, which is actually kind of interesting because researchers are there. The conversation is happening, but it's mostly within the social networks of the researchers. So so someone publishes a preprint, and instantly they start talking about it on Twitter, and that's where the conversation happens. And I think that's interesting because I think the roots of this very like kind of discussion of our show comes from Microbiology Twitter Journal Club, which was also based on discussing microbiology on Twitter. So
absolutely, yeah. And I mean, I still look to Twitter too. Like the people that I followed back then and that I still continue to follow are sources of finding, right? Like networks of people that are highlighting different types of research.
Exactly. I mean, guess where I found this paper? I found this paper on Twitter. It's not just the hellscape of hatred. You get lots of interesting science being discussed there.
Yeah. I mean, you have to find those small communities, right? And then, yeah, anyways, I don't know if I get into Twitter, good and bad.
You have to find your niche in Twitter, I think, is a key thing. And
yeah. And still, you may not be safe from some roaming comments.
Well, yeah, it's very hard because everybody's on Twitter, and you can't figure out how everyone will interpret what a certain piece of research would say or mean. So you could post something, oh, this is an interesting piece of research, and then you can get taken up by lots of people who don't understand it. And I think the only way to do that is to talk more about research, to get more people aware of kind of.
I mean, I have this crazy thought that I think like, you know, Twitter was a tool that existed, and then that was like so exciting. Like when we were going through grad school, we both jumped onto Twitter because it was a great place to meet all these different academics and all this different information, fuse our personal lives, maybe even with our burgeoning scientific lives. And but I you know, that's just because it existed first. Like I'm thinking there's probably better moderated options that could exist, but like no one cares anymore because, you know, this is the big network and everyone's on it. But there are people, I think, that are thinking about these questions and what sort of online communities might be better for research communication.
Yeah, I bet there's like at least 6 or 7 people in Silicon Valley who are trying to figure out something that they can be the next. I mean, I've known people like who have been coming up with various alternatives for like discussing science openly, but using social media. So I think I've seen like at least 3 different like companies talking about sharing protocols, and I think nearly all of them have disappeared. But like, oh yeah, be a researcher. You can use your phone in the lab to write up your protocol and share it with people. And I think during my PhD, I think I was on like maybe 2 or 3 of those that have all disappeared along with all the data I put into them. So it's
there's many opportunities for success and failure in making better scientific communication, both between researchers and between researchers and the public. And preprints are definitely one of those tools, but they're not some golden goose of scientific communication. Like we have to recognize their limitations, I guess, is one of the thrusts of this paper.
Yes. So I think it's an interesting paper, and it's an interesting perspective. It's from people who are quite interested in open science. I think it's part by the Open Science Foundation. They put, and again, in keeping with their own principles, they put all of their data analysis on their platform. So
that's a great platform. That's the origin point of a lot of that reproducibility crisis research. I use it to put classes, actually. When I develop classes, I put it in that platform.
Yeah, excellent. So I think we're going to go on to the next paper, which is the total number and mass of SARS-CoV-2 virions. So,
yeah, I think this is a hilarious paper. It really made me laugh when I read the title of it. This is something common though that happens inside of some ecology fields and a lot of theoretical, like computational biology. People want to know the boundaries of something, right? Like what's the highest amount or what's the lowest amount? And then from those sort of large boundaries, you can try to generalize some principles or rules. I'm trying to think. They're like bioreference. There's something like biology by the numbers. If people search that online, like you'll find places that like how many of this exists, right? How many bacteria exist in the world? Those types of boundaries are helpful for maybe like conceptualizing like a large, like a zoomed out view of what's going on. Um, and so in this paper, they look at primate primate tissues that are infected with SARS-CoV-2 to get a sense of how many virions are there, and then they use math back of the envelope calculations, which they formalize in a good way to find out what would that mean in terms of all of the humans, given that lots of humans are infected, and given that the tissues in humans are these are the tissues that are infected by SARS-CoV-2. And they come up with a collective mass of zero point 0.1 to 10 kilograms of SARS-CoV-2 virions circulating in human hosts.
Like that's just around the world, I think, or like currently.
Yeah,
And they
exactly.
and like they estimate that each infected person carries an estimated 1 billion to 100 billion virions. So, but even with that, the total mass is no more than zero point 0.1 milligram. And
They're
yeah,
tiny, right? Like virions are very small. Multiple of them fit in a cell, right? They're inside the organelles of a cell. These are not large objects. Um, but they do take a biomass. They are proteins and nucleic acids, so they have that mass. And I guess maybe it's important to say what they then sort of use that information for. They sort of they use it to if they think about like the total amount of nucleic acid that exists, and so how much is basically how much is available to mutate at any one point. And it seems like they're trying to use this estimation to figure out an upper bound of mutation potential.
yeah. I mean, it's so. I mean, when I first saw this, I thought, okay, so Trivial Pursuit of Pate funded this study for a bizarre fact. How much does SARS-CoV-2 weigh? But the interesting thing I found here was actually figure out like because they created this model and to create this model, it's actually quite complicated because you have to figure out and estimate like where SARS-CoV-2 is going in the body, what's it doing? Like, kind of, is it just in the lungs? What I mean, do you
Mm-hmm.
count say SARS-CoV-2 RNA, or do you? I mean, how do you count? I mean, it's
Right.
quite, and it's interesting like finding where they find the model breaks because that kind of so in here they've kind of used it to identify knowledge gaps in the available literature. So kind of we don't know the amount of virion yield per infected cell. And the only studies that cover that are from 40 years ago that on a different like beta coronavirus, and that's what they
Uh-huh.
plugged into their numbers. And things like the measurements of mutation rate per nucleotide per cycle. I mean, again, these are sort of things that I guess we wouldn't really necessarily think about if it wasn't for kind of out of the box studies like this.
Yeah, I mean, yeah, I guess what you're saying is in the pursuit of this question,
Yeah.
there's they look to the literature and say, okay, well, like we need to base this on some estimation that we see out there, but that process is a scientific process and saying, oh, we don't see it out there. So maybe that's a place that we should know answers if we want to figure out something like this.
Yeah, because I think yes, in this kind of study, you can it theoretically requires you to read pretty much everything there is to know about SARS-CoV-2 in order
Mm.
to figure out this question. So with that kind of and plug it into this model and figure out like where the model starts to break or where it. I mean, another criticism would be like no validation, but I don't think this was like kind of very intellectual exercise. Um,
Yeah, and that's sort of how I prefaced it too, right? I think that these types of papers come. I've seen these papers before in sort of ecology, computational, like theoretical, right? Like there's a lot. It's like a theoretical biology field. How many genes exist in all these different bacteria? Some comparative biology as well, because it gives you numbers that you can compare to other things, right? You can get a sense of where a problem sits in physics basically, right? Everything from 0 to 1, it helps you fit in there. Actually, that reminds me a lot of when we did the paper about how bacteria are behaving in this different, like this weird single particle phase transition way, right? And they had a
right,
graph that they put all the different sizes of tiny particles and all the speeds that they could possibly move, right? And physics does that, right? Like,
right.
and this type of paper is a little bit of a bridge to those types of analyses where you're just trying to zoom out as far as you can, and yeah, describe a system.
Yeah, so I guess going to the clickbait title that I used for this, if someone asks you how much what is the mass of SARS-CoV-2 variants in a person, you can say at the peak of infection, no more than 0.1 milligrams, and across the whole world between 10 kilograms and 0.1 kilograms. So
Yes.
when it comes up in a really twisted Trivial Pursuit question in 5 years' time, before you write a complaint to Trivial Pursuit, you can at least nail them with the right answer.
It's a pub trivia for sure, right? Like
Yeah.
this is trivia night at the pub.
Yeah, this is the kind of thing that will get either you know the answer or you spend ages trying to like feverishly go, okay, how much does the virus weigh? It's this amount of, and this paper will save you that.
Okay, what do we got next on the docket?
Next one is limited within host diversity and tight transmission bottlenecks limit SARS-CoV-2 evolution in acutely infected individuals. So this is a preprint. It's got a little yellow disclaimer. So that's how you know. I feel like now I have to be a bit more clear about it.
Yep.
Now this is interesting because this attempts to kind of nibble at the question of where do variants evolve. So we talk about variants a lot on this like kind of broadcast.
Mm-hmm.
And
I mean, all the news media I feel is
pretty
talking
much,
about variants
yeah.
a lot.
So I think one of the things we've touched on in previous things is this idea that variants evolve during prolonged infection. So when people have a SARS-CoV-2 infection for a very long time, that's where that gives the virus space to evolve multiple mutations and thus develop into a new variant. This looks at acutely infected individuals. It looks at the counterpoint to that. Because I guess this goes back to the whole like kind of how do you prove something? You try to look at the counterpoint to it and see whether that can actually be. So the idea here.
I'll just I'll lob in here that in the times that we've talked about evolution happening right within host evolution, I think a lot of the case studies we've looked at are in immunocompromised individuals,
Yes.
right? So these were like long-term chronic infections in immunocompromised people. This was just where the data existed, right? These were the opportunities that scientists had to glimpse, right? Like a population of viruses in a person. And you can imagine it's easier to do it in that case versus in an acute case, because in an acute case, like that's pretty short, right? Like you'd have to sequence very deeply to find the variants because there's probably not as many of them. You don't have as much sample volume. So yeah, this is also just a type of research that would take a bit longer than inside of the chronically infected individuals.
Yeah, exactly. So in this one, they take about 133 SARS-CoV-2 samples collected from acutely infected individuals in Wisconsin. And they look at the patterns of these things called intra-host single nucleotide variants. So nucleotide variants that evolve during the infection that have like just maybe one or two like kind of base pairs out of position, and see like whether any of these variants like stick, whether they change. And this is also like people sampled within a network of transmission. So they were able to look at people who they transmitted to see whether the mutations were carried on to the person they transmitted to. And basically what they found was that while they spotted some variants evolving, they found that they didn't transmit very well. So there was a big transmission bottleneck because the ability to transmit is a complicated process. So if you just break one part of it, it's more likely to induce a selective like barrier rather than to. So you have to long term. The idea I think is long term infection gives the time for multiple kind of mutations to evolve.
But at the same time, like at that point of transmission, how many virions are really coming from that pool of mutated things into the new host, right?
Yeah.
I think that's the point that they're trying to drive home in this paper.
Yeah. And I think, and a lot of this paper is dedicated to quality control because papers have been published like this before. But I think the key thing is that these kind of like mutations can evolve, can happen during the kind of sampling process of when you do a PCR. And
for sure.
So quite a lot of this paper is dedicated to making sure that they got everything technically correct, which is the best kind of correct. And
it's an important one for this. Yeah, it's an important one to claim that these variants have some significance, right? Because inside of all of our techniques for reading DNA and stuff like this, there are small error rates. And so if we're trying to observe small error rates inside of a target population of genomes, SARS-CoV-2 genomes in this case within a single individual, then we have to be hyper aware of all the different ways that the processes introduce small amounts of error as well.
Yeah, that's right. And I think so. This is quite an interesting study to look at, even though what they have is essentially a negative result,
Mm-hmm.
which is less exciting. But I think the way that they ask the question is quite interesting, and the kind of techniques they use to make sure that. So that's why I think this would be
Yeah.
kind of worth the read.
I
But
mean, it would be interesting too because, right, like I think maybe this is a nice chance to talk about how, like, what am I learning from just skimming it here, right? Is that I'm learning that there's this potentially there's support for a thought that the bottleneck of transmission is very tight, right? And that seems to make sense with most of the things that I already know, but I can't say for sure. So reading this paper would help me feel more confident in that, and maybe like where I would say like, yeah, that's something that I know, and use it in like the way that I explain the world. But right now it still sits in this kind of like, I'm kind of uncertain sort of zone. Yeah, and
yeah.
I guess that's preprints for you, right? Like
Mm.
they're interesting ideas, but until you dig into it or until you know someone else has dug into it to cross-reference that information properly, then they should sit in like a tentative, like maybe kind of
Yeah.
way of thinking about it.
So yeah, this is like uncertain information. Like your mate in the pub told you this. It sounds reasonable, but you want to make sure that there's other confirmation.
Yeah. Okay, cool.
Cool. So yeah. So you got another preprint called "The Importance of Time Post-Vaccination in Determining a Decrease in Vaccine Efficacy Against SARS-CoV-2 Variants of Concern." So this paper caught my attention because it's basically like dunking on a lot of other studies, basically saying like, oh, because there have been a lot of studies saying that variants of concern have been breaking through in infections, or they're going, and I think this paper goes in and says, okay, we have to look into the methodology of how this is being discovered. And they basically did some modeling, and they believe that a lot of the kind of variation is due to the fact that people are being sampled at different times after vaccination. And the thing is, after vaccination, it takes some time for that immunity to build up, which means that sometimes you might be sampling people in a position where they're more vulnerable to just being generally infected by SARS-CoV-2 than other. So they basically kind of propose that if we're going to be looking at whether any of these SARS-CoV-2 variants are escape mutants, we have to take into account the time after vaccination.
Yeah, so they don't have data, right, from these. It's not like they reanalyze the data in papers. It's more that they make an example. They call it a toy model, it
Yeah.
seems, where they say, imagine this case, right, which we know to be true,
Mm.
that immunity takes some time to develop. If studies sampled at this point, they would get a result that was influenced by that, right, that building immunity. And so then this is a reason to be critical of looking at some of these papers if they're not controlling for this factor.
Yeah, I mean, because I guess for the original version of SARS-CoV-2, we're thinking of in terms of a 2-week time to neutralization. Like, so I got the vaccine, so I got my second vaccine on like Friday, and so I'll be waiting 2 weeks. But actually, when you got variants of concern that might be slightly more resistant to antibodies, that timing might be like something like 3 weeks, or it might be slightly longer because more kind of more neutralizing antibodies are required. So it might take a little
Sure.
bit more time to build up that immunity, and that's an interesting thought.
Yeah, but it would be interesting. Yeah, I like, once again, this is talking about. I feel like the model is based on neutralizing antibody titers,
Yeah,
which
which
it's
yeah,
like some sort of we know it's a correlate. Yeah, it's some sort of correlate with general immunity, but we don't. I don't think the relationship is super well. It's not like it's certainly not the gold standard to say, but that's how they. But that is how they talk about efficacy in phase 1 and phase 2 trials.
I think you actually hit on a really important point there that we will be picking up in later papers we're discussing today. But yeah,
Yeah.
I think you're absolutely right because most of the things we measure are based on antibodies. So so I myself have fallen guilty to the myopia because I'm the one who suggested this paper and of like antibodies are everything. Antibodies, but of course the immune system is much bigger and much more complicated than that. There's a lot of other stuff out there other than the antibodies. We're just using antibodies
Right.
because they're the easiest thing to measure. but they're
Yeah,
not.
and it has tracked in our experience, right? If we think
Yeah,
about the phase 1, phase 2, and phase 3 trials that we've looked at, right, we saw good antibody response in phase 1 and phase 2, and that's played out as good clinical protection in phase 3. But the actual link between that, there's also the T cells going on. The B cells are the antibodies, but the cellular immunity
yeah.
set of things. So
Yeah, it's a good
okay.
marker of a good immune response, but if we just look at that marker
Hmm.
and think that that's the only thing, then we lose sight of all the things that it's actually indicating. So we're looking at our ruler going,
Hmm.
this is a great
Hmm.
ruler
Hmm.
measuring this length, but actually
Yeah,
the
yeah.
analogy breaks down there. Sorry, I don't know where I was going with that, but pipe up in the comments
Okay,
if you have a better analogy. All right,
let's
so
see what we got next.
next is ChAdOx1 vectored vaccine. AZD2816 induces strong immunogenicity against SARS-CoV-2 B.1.1.7 and other variants of concern in preclinical studies. So, AstraZeneca have developed a new vaccine targeting the what we're calling the beta variant, also known as the B1351 variant, VOC. They have many names. It's like Anna Karenina up in here. We got loads of names for these things. But yeah, this is exciting because we've been talking because various manufacturers have been talking about producing a new version of these vaccines that can protect against the new variants. And this is, I think, the first paper I've come across that really looks into it.
Hmm.
So they tested this out in mice, and they don't just look at yeah, they do things slightly differently here because they also look at whether it works if you give the original uh, AstraZeneca vaccine and then add it on as a booster. So
Mm.
so whether it's the second booster or whether you got fully vaccinated mice and then add it as a third booster.
Hmm. That's
So
interesting. I mean,
yeah,
that's like I'm sure that this is also built with some thought of all the different ways that having another version of the AstraZeneca vaccine could impact current vaccine dosing strategies out there, right? Like this paper is supposed to support a lot of different lines of thought.
yeah. So yeah,
Hmm.
they're looking at say, okay, what if we put it out tomorrow and lots of people already had their first dose of vaccine? Could this be used as a second dose? But they're also looking
Right.
even further ahead to next year and going, okay, let's say
Could this
we
be the
next
booster?
yeah, could this be another
Hmm.
booster? So, uh, but I mean, there are some flaws to it. It is, I mean, firstly, it's only done in mice, and they
Hmm.
don't, and because it's done in mice, they don't even bother checking whether it protects against real SARS-CoV-2. It's all antibody
Yes.
tests. So again, that's a
But
pro.
no, actually, it's more than just they do like some cell stuff, right?
Oh
They
yeah,
do
you're right.
interferon staining. So like they don't look at actual infection, I guess is what they say, but they
Mm.
do an in-depth characterization of the mouse's immune system's response to the virus.
Yes, they do that. They test a number of other variants other than the beta variant.
Hmm.
They have the Kappa variant, which is the Kent variant, the B11, the oh no,
Okay,
I
no,
sorry, I'm getting confused. Yeah, they test out against the
and
Indian variants.
the type virus as well.
Yeah, and the
Hmm.
yeah. So it's interesting paper. It's I mean it's not quite at the level we'd want to see if we want this vaccine tomorrow because we really want to see it done in
Oh yeah.
people and in a or at least in some kind of challenge model in hamsters perhaps or ferrets. But
Hmm.
this is a good first start, and so I thought
Yeah,
it'd
I mean
be
they say it's preclinical work, so they're
yeah.
not trying to claim that it's too much. But yeah, it's like quite interesting to. I think it's also nice to see um, how they've chosen their assays based on what we know now,
Hmm.
right? Like the SARS-CoV-2 field in some ways is very well developed. I think we've said this like a couple months
Yeah.
ago, right? Like there's enough information now that this is very much a mature field that chooses chooses specific assays that give us a nice rounded picture of what's going on because so many other papers have come before, right, validating some of these assays or like showing what features are important in disease, what you can look for, what are markers in the mouse of these types of things. Yeah, it's cool to see something, yeah, like the next step almost of the technological evolution of the
Hmm.
vaccine, yeah, in a paper.
Yeah. Speaking of
Another preprint.
yeah, another preprint. Speaking of preprints and evolution, we've got evolution of enhanced immune evasion by the SARS-CoV-2 B.1.1.7 variant. Uh,
Hmm.
so
So this paper we talked a little bit about, I think, NSP1 before, non-structural protein 1. That there's a whole bunch of non-structural proteins inside of SARS-CoV-2 that what scientists think are they're immune modulators of some sort, or they perform some function inside of the cell to make sure that the virus can find its niche intracellularly. And here they track down nucleotide changes inside of the variants in this region, and then test those changes test those changes in sort of cell culture models to find that some of those mutations are making NSP better at suppressing the immune response of cells.
I mean, I found this quite interesting because it talks a lot about those non-structural proteins, which is a big thing because whenever anyone talks about a new variant, they often focus in on spike and the mutations on spike and how those mutations are what will define what will happen with this new variant. But actually, we've been finding quite recently that those aren't necessarily true. I mean, in most recent example is in one of the multiple Indian variants. There was one of those variants had this thing called the double mutation, which was this set of mutations which made it less able to be neutralized by antibodies by, like, say, Regeneron or other studies. So there was a lot of panic about this double mutant that turned out to be it turned out not to be as dangerous as the single mutant. Because SARS-CoV-2 has a lot more going on than just the spike protein, and this really focuses that down. It shows that actually there are all these other evolution stuff that happens that could be affecting its transmission. In this case, they kind of show that these non-structural proteins are evolving at the same time, and that is also having an effect on immune evasion and escape. So there are other things that we might be looking at to see what's going to happen. I just realized,
I think maybe I kind of messed this up with the different papers. There are two papers like this that we have in the docket. This one talks more about ORF, or like they're non-structural proteins. ORF, where are the two that they look at here? ORF9.
ORF9 and ORF6.
Yeah.
So, yeah. But again, I think that other paper is going to feed into this. It's coming a little bit later in the docket, so stay tuned if you want to hear about what we're... But I think this paper really speaks to your earlier point about the overestimation of antibodies in SARS-CoV-2.
For sure, yeah. Like, it's not like the danger of the variant may not just be that it's not recognized by an antibody, right? Like, antibodies are just an indicator of what's going on. We also, the viral fitness inside the cell matters a lot as well, and those are things that we're not really measuring on a broad scale in terms of how people are stacking up against the virus in those respects, against the variants, right? Like, it would be interesting to see, like, a study, like, I'm just thinking now, this is kind of tangential, but it's related to this. What sort of data set would we want to see to tell us about how the variants have different immune abilities, right? We might want to see like T cell counts from people infected with with the different variants versus the wild types. We could say, oh, their immune systems are responding differently, but it's like super complicated to disentangle that and attribute that to the fact that they were infected by different variants, because we also have variation in our immune systems. So tracking down and like assigning that to the variant, it would be difficult. Yeah. Anyways.
Yeah. So, I mean, this one looks on syncytia. Well, so the thing is, like, when you've got like SARS-CoV-2 proteins in like kind of the cytoplasm, I think that they can allow cells to fuse together, and they can spread between cells. So I think lots of viruses can do this. So I know HIV and flu have ways of spreading between cells without actually escaping. So if there are two cells next to each other, they can just punch their way through into the next cell.
Oh yeah, sorry. I was just talking about some random tangent from the last paper. I wasn't thinking about this syncytia one.
Oh right, sorry. Yeah. So
yeah, but yeah. So this paper is about syncytia, respiratory syncytial virus. That's the first thing we learned that in like our first virology lab or something like that.
Yeah.
So yeah, some viruses are able to fuse cells together. That's because envelope viruses are using parts of the cell membrane, right, to coat themselves, and then those those particles go on to fuse with other membranes. So you can imagine, even before the virus buds out, that membrane is being activated by all these different types of viral proteins. So if it was sitting right beside another membrane, it could just fuse instead of having a budded viral particle fuse to another cell.
Yeah, and I think this is something that people have been seeing in SARS-CoV-2 outbreaks, where if you put some lungs on the microscope, there are respiratory syncytia that are being formed. And this
Oh,
study,
you've seen that? That's actually what I was going to ask. Because I saw another paper about syncytia, and I was like, oh, is this just a toy to experiment inside of cell culture, or is it something there's actual observation for in disease?
so I saw that there was a citation for a paper on that, so I think I
Nice.
would need. So I saw there was a citation to it, and I realized I haven't done my due diligence of checking that citation. So,
Okay, no, that's
uh,
fine. I just want to know, like, I guess that would be the interesting thing. Maybe next week we can put it on the docket or
yeah, I
something,
think next week
because it would be,
maybe. Yeah.
yeah, it would be nice to like. I've seen this talk about syncytia, and I was like, I didn't think it fit into the disease. I thought it was just when I was reading these headlines and abstracts, I was like, oh, this is just because people study syncytia, not because syncytia happen in disease. But if it does happen in disease, then it takes on greater significance, right? Because that would mean that this could be a disease process of some sort.
Right, yeah, for sure. But yeah, I think I need to double check that because again, this is a preprint, so but I think in this study they do test things in a cell culture, but again, it's
Mhm.
they create their own cell line to just so they mix up some lung carcinoma cells with some I think cells expressing certain proteins and
Mhm.
ACE2 receptors, and then was selected whether to see whether they're infectable with SARS-CoV-2, and then they mixed them together, and in their model they do take some videos of things of SARS-CoV-2 spreading between cells. Um,
Mhm. Yeah, and I guess it's interesting just that I don't know, the title gets me like insensitive to antibody neutralization. I would think, of course,
yeah,
it also matters like how many antibodies in this scenario. Like this is why like I want to know the role of syncytia in disease because I mean if it's like just like a few events, like you'll never really have that many antibodies, and
mm,
also then it's not antibody neutralization that I think you would want to inhibit this. You'd want like those cells to be identified by cellular mediated
yeah,
immunity and kill off those cells.
yeah,
Um,
because antibody can't really do anything against cells. This has to be.
yeah.
I mean, the thing about antibodies is they do limit systemic spread. So with cell to
Mhm.
cell spread, that's going to be limited into a certain like kind of foci of because it'll take because I mean it can occur rapidly, but can it occur more rapidly than if SARS-CoV goes from the blood to another organ? So
Right.
I think
Right.
that there is still like a reason why like because I mean there's still a reason why say treatments like Regeneron and stuff might be useful in last stages. I mean I think this paper really points out that maybe they're not a reason for why those convalescent serum might not be as effective as we hope. But
Hmm.
I think that there is
Hmm.
still an argument there, especially like earlier on in the infection. But this is an ongoing discussion that's happening in the field and with real doctors who are talking about this. So
Yeah.
uh, yeah, I
Yeah.
found this.
Interesting. It's an
Yeah,
interesting study. I'm glad that we talked about syncytia here because yeah, it is a way that viruses are studied in some other viruses.
yeah,
So to see it in SARS-CoV-2 closes the gap, I guess, a little, attaches it to even more literature out there. As if SARS-CoV-2 hasn't infiltrated every corner of the scientific literature.
true. Uh, and we've got another SARS-CoV-2. So our next paper is SARS-CoV-2 uses a multi-pronged strategy to impede host protein synthesis. So I believe this
So
is what
this
you
is
were
the
touching
one I
on.
think was the
Yeah.
NSP. Yeah, is the NSP one. And I was gonna say that this is. I almost see this as a follow-up study from. I think we like read something a while back where they did like a RNA-seq screen to find SARS-CoV-2 proteins that bind to RNA.
Yeah.
And then they identified, oh, it seems to impede protein synthesis, and they even talked about how like the SARS-CoV-2 mRNA has like a different sequence at the beginning that prevents its own proteins from suppressing its translation.
Yeah.
And so this, I think, is sort of the big follow-up study to that. Um,
Yeah.
to yeah.
I completely agree with that. I mean, we
Hmm.
also I think we touched on this because we did the replication organelle a couple times. We've done a couple of
Yes,
things
that's
on
right.
yeah. Um, but
Yeah, so this is a more in-depth view into that to get closer to the mechanism because like the paper that we looked at before, that was just a screen and then an example of a potential mechanism. Here they're going from, okay, like we know that there's a potential mechanism here, let's investigate that further and figure out how exactly is it suppressing the translation here. And maybe I think they also show that it suppresses certain types of transcripts, right? They can sequence like what is being suppressed. And if you remember from the last one, I mean, this is a long time ago now,
yeah,
but I think they were going off this theory of like interferon stimulated genes. So
right.
I'd be interested to see if this paper can replicate those findings, maybe in not like such a toy example where they specifically were inducing interferon stimulating genes and then seeing that they could get knocked down, but saying like, are the interferon stimulating genes in relevant cell types being suppressed?
Yeah, I mean, this looks goes into like the degradation of cellular mRNAs and kind of compares that to like how the cell treats its own mRNAs compared to the viral mRNAs during an infection, and
Hmm.
how that might
Yep.
change during infection, which is there's a lot of stuff in there that's quite interesting. Um,
Yeah, it's definitely a more detailed view of a very specific life cycle stage of the virus and that strategy in this life cycle stage that the virus is using to, gain dominance. Because we always say viruses hijack, right, your cells and make them into factories for themselves. But how exactly is that hijacking accomplished? And this is one
right.
of those mechanistic papers that is trying to explain one of the ways that that could potentially happen.
Yeah.
Okay.
The next paper, deep spatial profiling of human COVID-19 brains reveals neuroinflammation with distinct microanatomical microglia T cell
Hmm.
interactions. So, so a couple of weeks ago, one of our commenters sent me a study on COVID-19 entering the brain, but it was behind a paywall, so we couldn't really talk about it. So,
Hmm.
but eventually, but that was on my radar. So eventually, we found like this study that's open access, which talks about the spread of SARS-CoV-2 to the brain. So,
Hmm.
we did look at a study in hamsters a long time ago, which, as a tangent, also found that SARS-CoV-2 was specifically like entering the nerves in that region.
Yeah,
And
I remember seeing that you could see them, uh, the it was part of the smell, right? Like it could go
yeah,
here, but then it can travel back through those neurons then and get into some brain regions.
the olfactory bulb. I think
Yes.
I've mentioned that in my previous research that the olfactory bulb is quite pressed. So I worked on a nasal transmission model where I also found this, the disease I was working on spread to the olfactory bulb,
Mm.
and because one of the things that happens during nasal infection is that there's this little like kind of your nose like runs underneath your brain, and this organ called the olfactory bulb like sends in nerves and for your sense of smell, and it's
Hmm.
separated by this I think thing called the cribriform plate, which during really severe infections in mice I found at least could get punctured and allow things to go into that this
Mm.
area of the brain, and
Hmm.
so there
Like
is like
it's the gasket, the
yeah.
gasket around the nerves that are coming from the brain to the outside.
Yeah, and I think this study looks into that in more detail because humans have a very hard to sample olfactory, so it's very hard to spot this happening in humans because
Mm.
sampling sticking a needle into someone's brain via the nose
Brain
is
biopsy.
really yeah it's 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 not something that will get you a Nobel Prize these days. But yeah,
And
it's
not a study that can be done just so willy-nilly, right? Like
yeah.
there should be some less invasive measures of data collection investigated, or like a really like strong reason for why you have to take this specific type of biopsy.
Yeah, but I guess for COVID there are neurological symptoms that people need which we want to understand more of. So
Mhm.
anosmia, the loss of sense of smell, and then all the symptoms associated with long COVID that we're still trying to understand that are linked to the brain. So the question is, how is SARS-CoV-2 getting to the brain, and what is it doing there, or is it causing some aspect of the immune system to turn against the brain? So this actually
But yeah,
looks
I was gonna say in this paper they're talking about microglia. So
yeah.
this idea that there are the brain, right? Everyone talks about the blood-brain barrier, right? It like prevents certain things from going across. The brain then has its own resident immune cells
Mm,
in some ways that I think have crosstalk. I don't know so much about this if they have crosstalk with the systemic immune system. But yeah, they have their own immune system. So it seems like this paper is investigating its response.
Yeah, so they take sections from 25 COVID-19 patients who had succumbed to the disease and compare them with various control patients. So these are all people who have like died of the disease, and
Mm.
so by nature they had a very severe version of SARS-CoV-2, but that also meant that they could sample and get a lot
Yeah.
of detail about what SARS-CoV-2 were doing. So I think we had a very similar study about B cells, and I think one of the more interesting studies we did a year ago, where looking at B cells maturing in germinal centers. But this is kind of very
Yes.
similar to that, where there's something people who had died of COVID to figure out what had happened, and
Mm.
it's very interesting because they look at say viral antigen in the brain cells. Where is the viral antigen coming from, and what does that tell us about how SARS-CoV-2 is spreading to the brain? But
Hmm.
also about like what the immune system is doing in the brain and how that might differ from people who are dying of different things. So is there like a specific thing that COVID-19 is doing in the brain that we aren't seeing in other diseases?
Hmm.
So that
Oh.
yeah. Next one is symptoms and functional impairment assessed 8 months after mild COVID-19 amongst health workers.
Oh yeah, this is like a really short paper that I picked out just because we've been talking a lot about long COVID things and how there's not a lot of data on it. So I thought this seemed like a short little table basically that did follow up on a bunch of healthcare workers, and like it showed that loss of smell persisted for a really really long time
Mm.
actually. In these individuals, as well as a host of other neurological symptoms. They did like a quality of life assessment, like how is it easy to do certain tasks, and yeah, there was impact in that arena as well. So yeah, sort of a small data tidbit here. I don't think we'll cover it at all, but I just wanted to point out for people, yeah, that
Yeah.
there's more evidence for these long-term sequelae.
Yeah, even after mild COVID as well. That's another kind of
Hmm.
aspect that is interesting. Yeah.
Right.
And so I think that brings us to the end of our SARS-CoV-2 papers, and now we're going to be moving on to the other half of our news story. We talk about other interesting microbiology news stories, but I think this next one is quite an interesting one because it's not necessarily news, but I think it's quite timely and important for people to read.
Yeah, I was watching a H bomber guy video about vaccines and just vaccine hesitancy, and I hadn't heard this story. So maybe people are familiar with that there was this doctor, Andrew Wakefield, and he published a paper saying that there's this link that doesn't exist between vaccines and autism. And it got debunked, but of course, it spawned a whole movement it's the most recent piece of scientific evidence retracted now. It's the most recent piece of scientific evidence that anti-vaccine proponents latched onto. And so I didn't know this story, but there was an investigative journalist that spent like several years of his life going into this story and figuring out like what went into it. And many different venues. One of them is the BMJ here, so that's why I picked up this paper because we've read things from the BMJ before. It's kind of like a nice bridge journalist versus into the scientific literature. Just speaking about how economic factors, the desire to further one's own economic interests in the world was ultimately the driver for the publication of that paper. And yeah, it's like the video I watched was like almost like an hour and 40 minutes long. Maybe people can skim this, or
Oh,
even just hearing me talk about it. Uh,
yeah. Who makes videos that are more than an hour long? Gosh, how I feel so seen right now. Thank you
um,
so much for being with us. If you're this part of this video, I have to thank you so much for staying with us for this time, because I mean,
yeah, we've been rambling quite a bit, I think, during
yeah.
this particular news segment. But yeah, just wanted to point this out for people. I think it's like very meaningful to see that social factors impact right public health in such important ways, and they have in the past, and people have even investigated all the different social factors that went into it, right? And so today, in our day and age, yeah, like it's still happening. It's good to be aware of, to understand things in context, and yeah, hopefully to do better the next time.
Yeah.
Hmm.
So next paper I got is Unified Route for Flavivirus Structures uncovers essential pocket factors and conserved across pathogenic viruses. So this is flaviviruses. You may know these from such diseases as yellow fever, dengue, Zika virus, and
Mm-hmm.
West Nile virus. So various viruses are named after locations in East Africa, because there's a Uganda Virus Research Institute there, and so they discovered a lot of viruses there. Lots of interesting history, but that's not what this is about. This is about structures of flaviviruses. So they basically just found a way to do some. So working with flaviviruses is very difficult because they're hemorrhagic viruses. You don't want to catch them, so working with the lab can be quite difficult. So they developed a strategy for taking an innocuous flavivirus, so one that only affects mosquitoes, and then giving it a surface to make it look like the other more dangerous flaviviruses, and then using like I think cryo-EM to solve their structures, and then so they look at a bunch of structures from a different sets of viruses, and they find like they compare them, and they find there's this like unique pocket factors that are not unique. Sorry, not the opposite of unique. Conserved pocket factors.
Common.
Common. So they're common to all of these pathogenic viruses. So it's a very structural paper. It has like lots of like very fancy 3D images, but it's interesting.
So this is almost like this is a pseudotype investigation in many ways, right? Like they're reconstructing the outer coat of these. Like these are proteins, right? So they're capsids.
Yeah.
They're reconstructing the capsids of these viruses, of these dangerous pathogenic viruses. But the payload inside is like, something that infects mosquitoes. So like we don't have to worry about what they've made here. But we want to learn about that outside coat because, of course, we know the outside determines a lot of different things about the virus, where it can get into, like what it fuses to, and so forth. And so the similarities between all these different, that's pretty cool.
Yeah. And they do compare it to the wild virus structures, and then they delve into a lot more detail, and so I think they get down to like they go like lower than 3.1 angstroms, which is very, very small measure of things, and they find this kind of, and structures are quite interesting in their own right. But I think that the fact they found this kind of unique, not unique, opposite of unique, conserved. I keep making that. This thing that is very similar across all these viruses that could potentially be a target for vaccines, which would be quite interesting.
Yeah. So it's good that you mentioned that resolution, because I was gonna make a joke too, like pocket factors. What are pocket factors? It sounds cute, right? Like I think this is talking about talking about a feature on the surface of the virus, some little a pocket, essentially a little invagination inside of the capsid where stuff can fit into, right? And like based on what fits into it, that's probably what drives its specificity. And so you need that resolution to see these types of small features on the surface. This is like as simple as like amino acid side chain rotations, right? Just like small little movements that make a big difference.
Yeah. So
cool.
I thought that was quite a neat paper. I quite like cryo-EM, so
it's a very structural paper. Lots of structure.
Very structural. Lots of pictures. Very, very colorful pictures.
Very colorful pictures.
So next paper, a Tn-seq screen of Streptococcus pneumoniae uncovers DNA repair as the major pathway for desiccation tolerance and transmission.
This one's a preprint too, and this is a pretty basic sort of bacterial genetics pathogenesis sort of, or I guess not pathogenesis here, but this is like a pretty basic. You have a question about what's this bacteria, it does a certain thing. You're like, why does it do this thing? How can it do this thing? What are the genes important for it? So you set up an experiment where I think I talked about this last time. This is the genetic approach where you break things systematically in the bacteria and you say, okay, we broke random things. And now because it's broken, we figure out these are important for creating the phenotype. And in this case, they're looking at desiccation tolerance and transmission because Streptococcus pneumoniae can sit on fomites. And, that's a route of nosocomial infection in the hospital.
So, your kitchen counters, your door handles. One of the things that they don't have very much of is very dry, not very many nutrients. So the ability for a bacteria to survive on them depends on its ability to cope with the fact that there's not very much water there. And so it's quite important. And they do some measurement. They do some actual transmission experiments with animals.
Yeah, so
Yeah,
I guess that is maybe like that's the very beginning of pathogenesis, right? Transmission.
right. Yeah, so they look at like these DNA and they found, and I mean this paper was quite interesting because they use this technique which where they sample mice by swabbing their noses against a petri dish, and you might think that what a silly technique. Yeah, guess what? I developed that model in my
Oh
PhD.
yeah.
Yeah, that
Nice.
was they're using a model that I developed ages and ages ago. I think I've still got like a poster or like kind of with like kind of like my initial like data on it somewhere.
Oh, that's
But
awesome.
so it's really awesome that I'm like, oh my god, I've even though I've left sciences a while ago, my my memes are still infecting it somehow, and
It's a good
people
thing,
are,
right? I mean, it's
yeah.
it's a very direct way of getting what's going on in the nose onto a
Oh
dish.
yeah, yeah.
Yep.
It came from a number of experiments of different. We tried all sorts of different strategies to try and sample mouse nose, like using like because mice noses are very small, so it's like looking. Okay, how about we use a best suture wire? We could because you can't get a swab small enough. And in the end, I was like, can we just like swab the nose of the mouse on the petri dish? And like it's the stupidest idea you've ever heard of, which is why no one else has ever suggested it.
Simple and sweet, I think.
Yeah, it's
Yeah.
simple and sweet. I mean, there are some problems with it, but I don't. But fortunately, they sidestep that. They only use it as like they basically say 2 days of consecutive swabbing is an indicator of it being positive, which
Hmm.
is basically the way we went with our work because sometimes you don't necessarily know whether it's in the nose. There's some sketchy
Yeah, there's
correlation sometimes.
there's variance, right? Because like you're
Yeah,
not getting that surface exactly. Like it's all about like I guess the the
yeah.
nose swabbing
If
technique on the agar.
Yeah, it depends on how much the mouse is breathing, how much you've like
Hmm.
whether you dilute your sample afterwards, because you can either get a lawn of bacteria, which is basically no numbers, you just know the bacteria is there. So it's I
Yeah.
know this
So in
this
our lab, it was much more difficult to get things out of the nose of the mice. They did a lot of like anesthesia and then right lavage
yeah
of the nasal
lavage.
turbinates. Yeah.
Yeah, that again that's because I mean if you're doing it over a long term period of time, you want to be careful because my supervisor found that was concerned about anesthesia affecting bacterial growth, and so wanted
Absolutely.
to
You
be
can drown your mice too, right? Like there's a lot of animal handling things that complicate long course infection there.
Yeah, you need. I mean, you can if you use too much, but if you use under a certain amount, it should be okay. But it's yeah. Again, you want to limit the amount of contact you have with your mice because that
Hmm.
if you're taking care of mice, you usually want to make sure that they're happy and they're
Hmm.
most happy when they're being left alone by you. So
Yeah. Good. Yes, good points.
sorry, I
Okay.
should yeah. Sorry, yeah. Weird tangent there, but I was like, oh my god.
No, that is
Yeah.
exciting. I mean, I love like I picked it because it like yeah, it's like something that this technique is used a lot, and I think that we haven't really talked about like a classic like
Yeah,
bacterial mutagenesis experiment. Yeah,
yeah.
I thought it was interesting.
Yeah, so that's good. So next one is experimental evolution-driven identification of Arabidopsis rhizophar competence genes in Pseudomonas protegens.
So there are tons of bacteria that live on the roots of different plants that they make up something called the rhizosphere.
Mm.
And in this, they they look at the lineages, I guess, of the different organisms that live in the rhizosphere of a particular model that they have, and find out that in these communities, they do a lot of gene swapping. So they find the way that these competence genes get activated.
Yeah, I've not really got much to add. I'm not very like au fait with this. All I can say is that this should theoretically be published in MBio, but I couldn't. But unfortunately, I couldn't find the website for this study.
Oh, so this is only yeah.
This
It took something happened with the American Society for Microbiology's web pages. Like they went to a new system of organizing their journals, and their DOIs seem broken.
Yeah, yeah, I noticed that exact same thing. It's one that it really annoyed
If you
me.
check our Zotero, you'll be able to at least open. I only did it right before we started the stream. I
Ugh.
like re-pulled this down because I was opening all the papers and I'm like, why is this one broken?
Yeah, it's
Yeah, the PDF is out there.
yeah. So yeah, the PDF is out there. You can find it and you can email the authors. I don't know. But yeah, no, this is interesting. But
Okay,
yeah,
let's see what
next
else we
one.
got here.
So
Okay, this is another MBio one.
but this one, yeah, the link worked. So
Okay.
well, the link didn't work,
And
but it's findable by Google.
right. And in this one, I actually think I brought this up a few news articles ago. It was another Mycoplasma mobile in MBio paper. They talked about the molecular mechanism, and it's the same news article. I just looked at it again. I'm like, oh, actually, there's another paper that came out where they actually have these high-speed AFM images of the movement. And I'm like, well, that might be more interesting to look at.
Yeah.
There's like interesting video here, plus that cute diagram of how they did it.
Yeah, you can see this little cute cell with a smile on its face. But yeah, we did cover this in our last we covered a related paper from the same group in our last episode, which was looking
Yeah,
uh,
just talking about that. I guess this tiny bacteria, they've found a lot more molecular information about it in terms of how it moves. And then here are some biophysical measurements that they were able to take to better characterize its movement.
yeah. So I think the last paper was very much on the structures of the kind
Hmm.
of the proteins or the things that form its molecular machinery, and this is a lot more
Yep.
on like how it's used. So we
Hmm.
get a lot more like traces of kind of movement. So it's
Yes. Yeah, seeing the particles move, measuring how fast. I've never heard of high-speed AFM before. So like in
no,
AFM,
yeah,
it's like they take a little probe. You saw in the picture. It's a little probe and it scans over the surface and it's basically it's so tiny at the tip that it's repelled by the electrostatic forces. And so it's almost I think of it as like a very, very delicate stylus on a record
record, yeah.
player. And it goes across and then it gets, but apparently there's some method in which that you can use AFM to actually see motion, which means it would have to scan incredibly fast over that surface. I don't know. That's just what I'm getting from the title.
But yeah, this
That's
is
why I'm interested in reading it.
you're right. This is like one of those brand new bizarre technologies that
Yeah.
yeah. I mean, it sounds. I mean,
Probably not brand new, but like brand new to us.
brand new to us. Yeah, sorry. Yeah,
Yeah,
things.
right.
That, I mean, the person who invented it 10 years ago was like, "How dare you?" But I mean, because it's usually like that. I like when something comes to microbiology or biology, it's usually been tested by physics or chemists first, engineers, and then it comes to us, and we're like, "Okay,
Yeah.
this is a brand new technology," and they're like, "Not really."
Never before done study.
Never.
Well, never before done on these types of samples, perhaps.
I mean, I think literally like a biologist walked past a physics lab and go, "Oh, so you got this nice thing," and it's like grab the physicist and go and it's like, "Okay, you're working for us now."
Totally.
Yeah. Okay.
Okay.
Next one. A bacterial symbiont protects honeybees from fungal disease. So,
Yeah, I love this is a cool story. I love the stories about how our symbiotes or symbiotic bacteria help out in different scenarios. So in this case, they find a organism that grows on bees, and they find that it is protective. It's very straightforward, actually. This paper.
yeah, I mean they start by looking at this the species called Bombella apis, and they look into seeing. So they find that so they got these like uh, these interesting images that are of bee larvae that they take from their own kind of store, and they grow them.
Hmm.
They let them go, and they give them the Bombella apis, and that or without it, and add the funguses that affect it, and they find that oh yeah, these bees survive, and so they got lots of
Yeah.
very like kind of potentially haunting images of bee larvae, but also like they kind of discover that it's not just the bacteria themselves; it produces some products that prevent. So they has these biosynthetic gene clusters that
Mm.
that produce these antifungal compounds that they isolate. So I found that was quite interesting.
Yeah, I mean, I just think working in different animal systems is always interesting to
Mm.
me. So, one of the things that's always been cool, uh, in my opinion, about microbiology is it's everywhere, right? Microbes are everywhere. So they influence all sorts of living systems. And one of the reasons I would want to read this is like, oh yeah, like let's talk about a bee system. Like people love
Yeah.
bees, and like how do we study microbes in bees? And what effects could microbes even have on bees? And so yeah, to hear that it also impacted some like you can see some gene cluster expression. Like this is not the be all end all of a mechanism, but it's an interesting story.
Yes, and that definitely. I mean, you're right. It's not the be all or end all.
Yeah,
Yeah,
I meant for that. No,
yeah,
I
yeah.
didn't, but
I
it's
know you
perfect.
didn't, but I couldn't resist.
All
uh,
right, what's next?
And next one, again, this goes into another thing for looking at kind of biological products, integrating genomics and metabolomics for scalable non-ribosomal peptide discovery. Now, this paper, at first, it seems like kind of the idea of it is they want to find biosynthetic gene clusters. So in the
Hmm.
previous study, they found one in Bombella that could produce antifungal compounds. This one, they're looking at
Hmm.
say just if you say take if you sequence a whole like kind of set of genomes, would you, without knowing what kind of the bacteria is, would you be able to predict whether they have biosynthetic genes, and also would you be able to predict what those genes are actually creating?
Hmm.
So
Oh yeah, that's interesting. Yeah, this is like yeah, this is so there's so many sequences of bacteria out there,
mm,
right? And so much that isn't annotated. So this is some sort of technique, it sounds like, for annotating better what's going on in these, yeah, or identifying at least areas of interest for investigating
yeah,
these set of genomes.
it goes even like so. Firstly, you want to find things that are biosynthetic gene clusters. So you look at things that are very similar to ones we know of. And then what they do as
Hmm.
the next step is they go, okay, well, what products do these genes? So when these gene clusters are formed, what products do they take on one end, and what they do to them, and they build out this kind of model of what these genes that they don't know what the structure are, these and what they're creating, and then they do mass spec on the other end, and go, okay,
To
we've got now got
verify if that's happening.
yeah, they have a
Hmm.
list of compounds that are created, and then they can try and predict which of these compounds are being created by this biosynthetic gene cluster.
That's yeah, that's again one of these interesting interfaces of taking lots of omics technologies and placing them together, right? The power of computation and sifting through very large data sets.
Yeah, so they identify the genes, they identify what they're doing, figure out how they line up in the assembly line, and sometimes there are multiple, and then take that and pair that to mass spectrum and try and figure out what is interesting. So they take some data from various, like the Tiny Earth Antibiotic Discovery Project is one of the things that I noted, but they also, because of this, they discover various like new non-ribosomal peptides. So I didn't really talk about this, but non-ribosomal peptides are peptides that are created outside of that system of ribosomal things. So they're created exclusive by
Hmm.
these biosynthetic gene clusters. So
Oh
important
wow,
things,
I didn't
yeah.
know that that was even a thing.
Oh yeah. So vancomycin, bacitracin, and tuxebaxin are all created
Oh
by
right.
these gene clusters. So
Yeah, I
also
forgot that they're like yeah, they're peptide based, so but they're not translated.
yeah.
Hmm.
So they get like structures you can't really do with a ribosome. Like I'm not sure exactly like what the details are, but a lot of
Hmm.
really important like products are created in this
Yep.
way, and so this is another way to find new antibiotics or things that
Cool.
could potentially be interesting. So or just new chemicals, which we don't know their function yet, but they could be interesting. And so they kind of find 121 previously unreported non-ribosomal peptides through this.
Hmm.
And yeah, it's kind of bonkers how they kind of can, because you just from what a genome and some mass spec dissect this entire kind of biosynthetic gene cluster that would have taken like multiple different experiments before.
Yeah, I mean all the knockouts and things that people would do and and like now you can just take these giant data sets and do them almost simultaneously. I'm sure like this is one of those things where you want to follow up and like be able to knock them out and to say the final word, but an amazing tool for getting to that information quickly.
Yeah, and the final paper we'll be talking about is co-evolutionary phage training leads to greater bacterial suppression and delays the evolution of phage resistance.
Yeah. So I'm not sure if people have heard about phage, what do we call it, phage
There.
therapy
Yep.
before. Yeah. Essentially, it's an old technology. I think it was people think of it as also from the Soviet Russia days. Like that's where it was primarily piloted.
Mm.
Um, a way of taking phages, like everything has viruses that affect it, even bacteria.
Mm.
And so you can take the viruses of bacteria and use that to kill bacteria. But of course, there's bacteria have an arms race going on, an evolutionary arms race with these phage to try to evolve immunity towards them because they don't want to die from random phage. But in this particular in this paper, they show a way of doing a coevolutionary experiment and finding a way to get phage cultures that are better at killing bacteria without creating resistance profiles.
Yeah, this is interesting because again, I mean, you mentioned the bacteria and phages, they're natural enemies, and bacteria have lots of
Yes.
defenses that they can evolve against them. I mean,
Hmm.
CRISPR is probably one of the most famous ones, where that is actually
Hmm.
originally, in its real form, a defense that bacteria use against phages. And
Yes. It's its native form, we'll say.
yeah, yes, in native form, that's right. And phage, because of this, we have this problem where like phages can, but bacteria can become resistant to phages very quickly. So like I think
Hmm.
they refer to a meta-analysis published in 2018 that says that 82% of animal gut decolonization studies develop have bacteria developing phage resistance, and like
Mm.
75% of clinical cases where they use those phages. So they do pair out that there is like a story here. And how coevolutionary training works is you basically take your phage and then you throw it up against bacteria and wait for that bacteria to develop resistance and then develop a new phage to counter that resistance. So you're basically trying to game out how the bacteria will evolve in the future and then prepare
Play
for
it, yeah,
that.
play it to its extreme until the bacteria doesn't have any space anymore. All right, the phage has found a target that is just the perfect, the perfect target that bacteria don't want to evolve away from.
Yeah.
Maybe it's
So
something critical, or yeah, there's many different pathways potentially for that.
yeah, so they conducted this with E. coli using either untrained or trained phages to evaluate the phage. Because another big concern about coevolutionary training is if you evolve a phage to a new strain, will it still be able to affect the old strain, i.e., the one that actually exists?
Mm.
So this
Mhm.
kind of works to try and address that question. Um, so yeah, it'd be interesting to delve in from this perspective. I think it'd be interesting to delve into the mechanisms of phage resistance. I mean, is it CRISPR or are there other things at play that we need to be aware of?
Sure, absolutely. I mean, yeah, I remember when I gave I feel like I've given a CRISPR presentation in the past, and I've seen these reviews about phage mutation because like it's interesting for biologists because it's an ecological system, right? There's predator and prey here. There's bacteria and phage, inside of natural environments. Yeah, quite a bit has been written on this. I like it as a springboard for that discussion. I agree, is what I'm saying.
Yeah,
I'll have to
I
say I agree.
Yeah, I mean, we haven't really talked about phages much. I don't think we've actually talked about phages at all in the year we've been doing this. So I think this will be
Yeah.
a good starting point. Because again, it's quite hard because lots of places don't really do much. There's not really much phage research happening. Um,
Hmm.
but partly because of this evolutionary kind of issue that's been happening. Um,
Well, I was just thinking about phages, like the when I the first time I learned about phages, I was doing some genetic engineering right in the lab, and I was talking about, oh, what do these strain names mean? Like, why is this BL21? Why is this whatever, right? And they're like, oh, you want to use this one because that's one of the resistance to phage. And then someone had a
mm.
horror story of like, one time I tried to grow up a liter of this and it was all gone because a phage destroyed it. So like now I'm always using the phage resistant strain. And after it went, like they had to clean everything so well because the phage
Mm.
was everywhere. So like people treat them like these like in some technological pursuits, phage are this pest, right? That's like, oh, they could like destroy a whole reactor's worth of bacteria. But we have to remember, they come from nature and they have this like evolutionary history. So people who study ecology, phage are like fascinating in some ways. Oh, we talked about it in the last news
Yeah,
episode
you're right.
too.
We
The
did.
phage of SAR 11. Yeah, we
Yeah.
never got. um, so I mean, we could. I'm all for doing phage in the next episode. Do you want to do this coevolutionary one, or do you want to do the SAR-11 phage?
I kind of want to do the co-evolutionary phage training because SAR-11 is very much in ecology, like how phages are important for the ecological setting. And as
Yeah.
someone who is a human, I'm interested in things that affect me personally, and therefore I want to see whether this can help when antibiotic resistance becomes more widespread.
Yeah, okay. I'm down. Let's do it. That made it easy. That was
Yeah,
a great segue into the paper that we would ultimately choose.
yeah, I'm totally up for that because I guess tune in if you're interested in antibiotic resistance and phages and whether could this be the new replacement for antibiotic resistance. Who knows? Maybe this paper will answer that. Maybe it won't. But we're gonna try and delve in to find out that answer next week.
Absolutely. So yeah, we want to remind everyone that while we're very enthusiastic about microbiology and somewhat qualified, it's possible that we didn't get everything right. Science is about thinking critically and asking the right questions. So if you have any questions or corrections, please let us know in the comments.
Yeah. Thank you. It's been a pleasure listening to you, Danny. And pleasure chatting to you as well. Um,
Same here, Fuzz.
so yeah, tune in next week for more microbiology content.